Two common chi-square tests that apply for data with two categorical variables are a test of homogeneity and a test of independence. Starting with the test of homogeneity, consider collecting data on the honeybees, bumblebees, and butterflies that visit a flower bed and analyzing the data to find out if the distributions of flower colors that are visited by these three kinds of insects are all the same. The data consist of counts of the numbers of honeybees, bumblebees, and butterflies observed visiting each of 4 different colors of flowers. The null hypothesis is that the distributions are all the same, and the alternative hypothesis is that they are not the same. To work out the counts that are expected under the null hypothesis, it is convenient to add to this table a row giving the total counts in each column and a column giving the total counts in each row. Entries in the additional row and column are commonly referred to as the marginal totals of the table. Here's an empty table to use for recording expected counts. To get started, consider calculating the expected number of honeybees that visit red flowers, which is the proportion of honeybees that visit red flowers multiplied by the total number of honeybees. Under the null hypothesis, these insects all visit red flowers in the same proportions, so the proportion of honeybees that visit red flowers can be estimated as the total number of visits by any type of insect to red flowers divided by the total number of entries in the table, or 26 divided by 107. The expected number of honeybees visiting red flowers is that proportion multiplied by the total number of honeybees, or 26 divided by 107 times 44. This result can also be calculated as the product of the marginal total for that row multiplied by the marginal total for that column divided by the total number of counts in the table. This same calculation can then be repeated to find all of the entries in the table of expected counts. The formula for the test statistic is much like the formula used in doing a chi-square goodness of fit test and is the sum over all events in the table of the square of expected counts minus observed counts divided by expected counts. All of the arithmetic to calculate this sum can be done on the computer. These inputs start with the observed counts, calculate the row and column totals, the total number of events in the table of expected counts, and the chi-square statistic. The sampling distribution of this statistic is well approximated by a chi-square distribution with degrees of freedom given by 1 less than the number of row categories multiplied by 1 less than the number of column categories. The p-value can be calculated as before using the cumulative density function CDF to get the area in the tail of the chi-square distribution with the appropriate number of degrees of freedom. This p-value is about 0.2%, so at a significance level of 5%, the conclusion is to reject the null hypothesis. A chi-square test of independence is a hypothesis test to detect correlation between two categorical variables measured for a single sample. For example, consider the following data showing blood type and eye color for a sample of 592 individuals and analyzing the data to find out if there is any correlation between blood type and eye color. The null hypothesis is that there is no correlation, and the alternative hypothesis is that there is a correlation. Calculation of the p-value for this hypothesis test is in fact done in exactly the same way as for the test of homogeneity. These inputs compute the p-value by starting with the observed counts from the data table and otherwise doing exactly the same calculation as in the previous example. The p-value in this example is more than 98%, so the conclusion is to fail to reject the null hypothesis. The difference between a test of homogeneity and a test of independence is not in the calculations, but in the type of data and in the question that is being answered. In a test of homogeneity, like the first example, there are 2 or more samples, and the question addressed by the hypothesis test is whether those samples were drawn from populations with the same distribution. In the flower bed example, there is a sample of honeybees, a sample of bumblebees, and a sample of butterflies, and the null hypothesis is that the distributions of flower colors that attract these different insects are all the same. A test of independence, like the eye color and blood type example, uses one sample with 2 categorical variables, in this case blood type and eye color, measured for each individual in the sample, and the question addressed by the hypothesis test is whether those measurements are independent. Since the test of independence and the test of homogeneity use exactly the same calculations, the distinction is not always important, but it is almost always useful in any analysis to be clear about the purpose of the analysis and the questions that are being addressed.